the next talk will be on organizational principles of semantic control in the human brain, and it will be presented by KAIXIANG ZHUANG from ISTBI.
Thanks to the kind introduction. Hello, everyone. My name is KAIXIANG ZHUANG from ISTBI Fudan University. And today I'm going to introduce my research study about brain's organizational principle of semantic control. So semantic information is crucial for assigning meanings to the objects and places around us and even our internal thought and feelings. However, readily available semantic information often falls short to meet the changing contextual demands of our environment. So for example, while bank is strongly associated with money, this association is inappropriate in the context of fishing. In such cases, we must engage in semantic control to assess less dominant part of our knowledge that better fit the current situation. In this case, river. And prior research has shown that higher semantic control reduced participants' accuracy and could result in longer reaction time during the semantic retrieval. And at a network level, the demands of semantic control have been associated with a specific network, including regions such as IFG and posterior MTG. However, previous findings, as summarized by the recent meta-analysis, have mainly focused on the selective dimensions of semantic cognition, such as concreteness and typicality of the words. So in addition, the precise neural correlates and semantic control and their organizational principles remain a topic of debate. So to this end, recent advances in language processing models offer a unified opportunity to model the semantic distance in a more continuous and multidimensional space that enable us to investigate how brain responds to varying semantic control demands in a more fine-grained way. And with this objective in mind, we employed a four-choice semantic retrieval task as 3T MRI, in which participants were presented with a keyword and asked to select the most conceptually relevant target word from two distractors. They were assigned to two groups to perform this task with no overlapping 80 trials provided for each group. Next, word embedding models were used to quantify the semantic distance of different retrieval trajectories. Applying this method, we obtained a continuous distribution of semantic distance, which we aim to predict using trial-level brain responses. For the preprocessing of neuroimaging data, we just use the legacy version of minimal ACV pipeline to pre-process it, and use the GM single to extract the single value beta maps in ICA fixed noise CFD data. Next, we apply the PI-based machine learning method of the cross-validation framework in order to test the accuracy and robustness of the whole brain features in predicting semantic distance. Here we refer to this predictive model as the semantic control signature. In order to provide a deeper understanding of this semantic control signature, we then combine methods such as brain postulation, meta-analysis, and RSA. And to begin with, the behavior results indicate that semantic space is largely shared across people. The split reliability of reaction time showed that participants' response speed across different trials were highly similar, suggesting that the position of the concepts within people's semantic spaces were largely aligned. Moreover, at the group level, reaction times could be consistently predicted by the semantic distance identified by different brain embedding models. As the semantic distance gradually increased, the reaction time of participants also increased, indicating a corresponding rise in semantic control signature. And although all models here showed acceptable performance, a graph knowledge-based brain embedding model called ConceptNet showed optimal performance consistently, and was thus applied in all the subsequent analysis. Additionally, we confirmed that participants' response were primarily influenced by the distance between the queue and target, rather than other potential factors in this task, such as the distance between queue and distractors. So, more importantly, our predictive predictive model revealed a highly stable semantic and control signature across 2 independent groups. The positive semantic control signature highlighted a left lateralized network that highly overlaps with the classic semantic control network previously mentioned in the literature, including regions such as IFG, posterior MTG, AVI, and dorsal medial PFC. These regions primarily belong to the language frontal parietal and dorsal attention networks as identified by the co-antispecific parcellation. And on the other hand, the negative signature was primarily composed of parts of the default mode networks, so thus the more automatic retrieval of very closely related words is linked to a stronger default mode activity. In addition, further investigation revealed that the semantic control signature can capture subtle differences of semantic distance across trials in a continuous manner. And we further show that the semantic control signature can outperform the language models in performing participants' reaction times, indicating that it may capture additional computational information about semantic control that is not accounted for by language models. We also demonstrate that semantic control signature can not only predict individual semantic distance within a certain group, but it can also be generalizable across groups. These results further confirm the accuracy and generalizability at both individual and group level. Having established a robust semantic control signature, we then further attempt to further interpret its potential cognitive significance. For that purpose, we employed an additional PIS model to link the linear combination of brain maps from the Neuroscience database with our semantic control signature. Here we revealed that 2 significant components that explain approximately 50% of the variance in this signature. The brain regions with the greatest contribution to the first component largely overlaps with the classic multiple domain network, which is associated with the cognitive terms commonly required for high levels of executive control demands, such as context, decision, and goals. On the other hand, the second component highlights the left lateralized brain network that largely overlaps with the core language network. So the cognitive terms associated with is just the commonly required for the semantic and language processing, such as meaning, reading, and comprehension. Interestingly, the weights of these brain maps were primarily concentrated with a specific interval along the principal connectivity gradient, situating the semantic control signature between the default mode and the multiple domain network regions. So this echoes with some recent findings suggesting that this layout may enable semantic control regions to integrate long-term conceptual knowledge with a more adaptive representation of the task-dependent goals. Now, we know that the key brain regions of semantic control signature are situated at the apex of 2 cognitive manifolds supported by several networks. So we then asked whether these regions may account for dissociable computations. For that purpose, we employed an RSA approach and calculated the representational similarity matrix of key brain regions across trials and examined the extent to which different brain regions share representational information. Next, we use UMAP to project the brain regions in a low-dimensional space according to the representational patterns using just during the performance of semantic retrieval. So our clustering results on the representational space indicate that language network regions are divided into 2 parts, each forming closely connected subsystems with the dorsal attention and the frontal parietal control networks, respectively. We also found that the representational connectivity of one of these systems was enhanced with increasing semantic control demands, which reflect the integration of the broadcast area with the regions of frontal parietal control networks. The other subsystem, however, just mainly reduced its variation without any significant increasing. Together, we suggest that these 2 subsystems may account for just dissociable computations during the semantic retrieval. In conclusion, our findings revealed that participants share a common semantic space which can be reliably modeled using NLP-based approach to investigate the neurosignature of semantic control. The identified neurosignature included regions associated with both language processing and executive control demands, which formed two dissociable subsystems. Future study will be required to assess the generalizability of these neurosignature to other different modalities and to investigate the potential functional differences between the identified subsystems. Finally, thank you very much to my mentor, Denis Vitansevich, our team members, and Beth, Johnny, and Phil. Thank you very much for your attention.
Okay, we have one question on the app. So could you please explain how did you optimize your sequence to get sufficient temporal resolution to be sensitive to the memory retrieval network alterations?
Would you mind you just repeat the question? Optimize what?
The question is, how did you optimize your sequence to get sufficient temporal resolution to be sensitive to the memory retrieval network alterations?
So we just use the latest, coming method called GM single, and these tools can help a lot in just decrease the noise in the fMRI data. So yes, that's what we use to optimize the sequence and for the prediction. Yes.
Great. Thank you for the presentation.
Thank you very much.